Now let's see how antiviral drugs will act on viruses. So because viruses are not alive, they cannot be killed. The only way for the antiviral drug to work is to interfere with any one of these processes. So either preventing the virus from attaching on the receptor, preventing the virus from injecting the genetic material, preventing the viruses from escaping. So these are different modes of action of antiviral drugs. And because we have different types of viruses, we have different types of antiviral drugs. And each type of antiviral drug targets a specific type of virus. So the first way is by binding to the cell receptors targeted by viruses or the capsid protein so that the virus cannot attach to the host cell. So one way is to prevent the virus from attaching here. So either the drug is going to target the protein, the capsid protein of the virus, or the drug is going to bind on the receptor sites of the cell. So the drug, let's say the drug is in blue. Yeah. So the drug will bind to the receptor sites targeted by the viruses so that the virus will not have any receptor site to even bind on. So this is one way that drugs can act. However, these drugs are slow. They are expensive, and so far they have not led to any commercial products. Another way for antiviral drugs to act is to inhibit the uncoating of the virus and preventing it from injecting its genetic material into the cell. So this is a second mode of action. So here, the drug is going to prevent the virus from even injecting this genetic material into the cell. So if no genetic material is injected into the cell, there is no replication or any of these processes will not happen. Right? So this is how amantadine and rimantadine act. They treat influenza and they use this strategy. However, most viral strains had developed resistance to these drugs. So viruses, as we've seen before, they mutate quickly and they adapt to drugs. So viruses have adapted to these drugs and these drugs are not effective anymore. The third way that antiviral drugs work is to mimic genetic material so that the virus cannot cause it to make copies of itself. Now these drugs, they will act on the host cell. So they will act here and they will mimic the genetic material of the host cell. But the genetic material of the host cell will become inactive, which means that even if the DNA or the RNA of the virus enters, they will not be able to give the host cell instructions to make copies of itself. This means that there is no replication of the virus. So if you read here, it says that viruses cannot use the genetic material of the host cell to multiply. The drug blocks the enzyme activity within the host cell and prevents the virus from multiplying. So if you do biology, you know that inside cells you have enzymes called DNA polymerase. DNA polymerase are enzymes that help to create DNA molecules by assembling nucleotides, which are the building blocks of DNA. So when this enzyme is altered, it becomes inactive. So now the DNA of the virus or any genetic material of the virus will not be able to be assembled. So that's how it works. Another mode of action of antiviral drugs, examples Tamiflu and Relenza, So these are called oseltamivir and zanamivir. These drugs, they prevent the virus copies from leaving the cells. So it acts in this stage. So it prevents this stage from happening. On the virus, there is an enzyme called neuraminidase, and this virus, it allows the different virus copies to be released from the host cell. So this drug will inhibit this virus here, will inhibit this enzyme here. And once this enzyme is inhibited, viruses are trapped inside the host cell. Therefore, it slows down the spread of the virus around the body. So all these steps will take place from A to E, but these viruses will stay trapped inside the cell. And this will only slow down the virus, and both of these drugs are used as antivirals to prevent the spread of the flu virus. This drug will allow the body enough time to fight the virus, and it will also allow the symptoms to be treated. So here you have a structure of oseltamivir, and here you have the structure of zanamivir. You need to know the different functional groups in these molecules. These molecules are available in the data booklet.